بسم الله الرحمن الرحيم. Dr. Ahmed Al-Najjar, Lecturer of Histology and Cell Biology Department. Today we will discuss and explain the nervous tissue. By the end of this lecture, the student will be able to 1. Classify nerve cells morphologically and functionally. 2. Describe the structure and correlated function of the nerve cell body, perikaryon, and processes including axon and dendrites. 3. Describe the structure of myelinated nerve fiber and classify the types of the nerve fibers. 4. Clinically correlated between defective myelin formation and multiple sclerosis. The nervous tissue is the fourth basic tissue of the body. What is the basic tissue of the body? Epithelial tissue, connective tissue, nervous tissue, and skeletal tissue. Anatomically, the nervous tissue is divided into central nervous system (CNS) and peripheral nervous system (PNS). The central nervous system composed of brain and spinal cord. Peripheral nervous system is composed of cranial and spinal nerve and their associated ganglia. Again, the nervous tissue is the fourth basic tissue of the body. Anatomically, the nervous tissue divided into CNS and PNS. CNS, brain and spinal cord. PNS, cranial nerve and spinal nerve and their associated ganglia. The nervous tissue histologically, it is formed of nerve cells and glial cells. Nerve cells, neuron, glial cells, neuroglia. The neuron is the structure and the functional unit of the nervous tissue. Neuron consists of two parts, cell body and cell processes. Cell body is called perikaryon or soma. Cell processes, axon or dendrites. Again, the neuron is the structure and the functional unit of the nervous tissue, which consists of two parts, cell body and cell processes. Cell body is called perikaryon or soma. Cell process may be axon or dendrites. Neuron is characterized by excitability and conductivity. Excitability, they respond to environmental changes by generation of the action potential or nerve impulse. Conductivity, they are capable of propagation of the nerve impulse to other neuron, muscles, and the gland. Again, neuron is characterized by excitability and conductivity. Excitability, they respond to environmental changes by generation of the action potential or nerve impulse. Conductivity, they are capable of propagation of the nerve impulse to other neuron, muscles, and glands. Neuron classify according to number of processes. Again, according to the number of the process, the nerve cells are classified to unipolar nerve cells, bipolar nerve cells, pseudo-unipolar nerve cells, multipolar nerve cells. Again, according to the number of the process, nerve cells are classified to unipolar, bipolar, pseudo-unipolar, multipolar cells. Unipolar nerve cells has one process. Pseudo-unipolar cells has one short process that soon divides into two processes, central and peripheral process. Bipolar nerve cells has two processes, central and peripheral. Multipolar nerve cells has multiple dendrites and one axon. Again, according to the number of the process, the neurons are classified to unipolar nerve cells, pseudo-unipolar nerve cells, bipolar nerve cells, multipolar nerve cells. Unipolar nerve cells has one process. Pseudo-unipolar nerve cells has one short process. Which divide into two processes, central and peripheral process. Bipolar nerve cells has two processes, central and peripheral. Multipolar nerve cells has multiple dendrites and one axon. Cell body pericardium is the part of neuron containing nucleus and cytoplasm. With light microscope, cytoplasm appear as basophilic with H and E and show nasal granules with toledim below. They are known as chromophile substance as they like stain. Nucleus single, large, rounded, central vesicular with prominent nucleolus. Owl eye appearance. Again, cell body pericardium is a part of the neuron containing the nucleus and the cytoplasm. With light microscope, cytoplasm appear basophilic with H and E. Show nasal granules with toledim below. They are known as the chromophile substance as they like stain. Nucleus, single nucleus, large, rounded, central vesicular with prominent nucleolus, owl eye appearance. Cell body pericardium with electron microscope, nasal bodies. Nasal bodies are aggregation of ribosomes, polyribosomes, or ER. They are known as chromophile substance as they like stain. Again, cell body with electron microscope showing nasal bodies. Nasal bodies is or are aggregation of ribosomes, polyribosomes, rough endoplasmic reticulum. They are known as chromophile substance as they like stain. Nasal body in all the cell body except axon and axon helix site of axon emerge from the cells. They disappear in nerve degeneration chromatolysis. Again, nasal body in all cell body except the axon and the axon helix. Site of axon emerging from the cells. They disappear in nerve degeneration chromatolysis. Cell body with electron microscope, the cytoplasm containing well-developed Golgi apparatus and a large number of mitochondria. Golgi apparatus sites perinuclear inside stain, sulfur stain. Again, cell body with electron microscope, the cytoplasm containing well-developed Golgi apparatus and a large number of mitochondria. Golgi apparatus site in perinuclear, staining with sulfur stain appear perinuclear. Cell body with electron microscope, cytoskeleton component, well-developed microtubules called neurotubules, intermediate filaments called neurofilaments, microfilaments, actin filaments. Again, cytoskeleton component of cell body with electron microscope appear well-developed microtubules, neurotubules, intermediate filaments, neurofilaments, microfilament, actin filament. This picture showing kinesin and dynein transport vesicles in nerve axon. Outward transport neurotransmitter uses kinesin. Inward transport recycled membrane use dynein. Neurotropic versus uses dynein as transport carrier. With EM, cell body absent centrioles in nerve cells can't divide or regenerate. Long-lived cell body with electron microscope showing absent centrioles. Nerve cells can't divide or regenerate. Long-lived. Inclusion: glycogen and lipid. Lipofuscin increase with age. Melanin, some neurons are pigmented as in substantia nigra. This picture showing melanin granules in the cytoplasm and showing substantia nigra, melanin pigmentation. Again, inclusion present in cytoplasm of cell body, glycogen and lipid, lipofuscin and melanin. Some neurons are pigmented as in substantia nigra. This picture showing melanin granules or substantia nigra around the nucleus in some neurons. Nerve cell process, axon and dendrites. This table comparison between axon and dendrites. Axon is single, dendrites are multiple. Axon long, thin, uniform diameter. Dendrites is short and thick and tapering. Axon It doesn't branch except at terminal end. Terminal arborization. Dendritis, extensive branching. Cytoplasm of axon containing mitochondria and cytoskeleton. No nasal granules, no Golgi. Cytoplasm of dendritis containing all organelles as a cell body, no Golgi appear in cytoplasm. Axon function. Carry impulse away from the cell. Centrifugal. Dendrites carry impulse towards the cell. Centripetal. Again, comparison between axon and dendrites. Axon is single, multiple. Dendritis is multiple process. Axon long and thin, uniform in diameter. Dendritis thick, tapering, short. Axon doesn't branch except at terminal end, terminal arborization. Dendritis extensive branching. Cytoplasm of axon containing mitochondria and cytoskeleton. No nasal granules, no Golgi. Cytoplasm of dendrites, all organelles as a cell body, no Golgi. Function of axon, carry impulse away from the cell, centrifugal. Dendrites function, carry impulse towards the cells, centripetal. Nerve fibers are the individual axons of nerve cells in a connective tissue sheath. Again, nerve fibers are the individual axons of nerve cells in a connective tissue sheath. Connective tissue covering of the nerve trunk are endoneurium, perineurium, and epineurium. Endoneurium, connective tissue sheath around individual nerve fiber. Perineurium, connective tissue sheath around each bundle of the fibers. Perineurium, epineurium, connective tissue sheath surrounding the whole nerve trunk. Again, connective tissue covering of the nerve trunk are endoneurium, perineurium, epineurium. Epineurium, connective tissue sheath surrounding the whole nerve trunk. Perineurium, connective tissue sheath around each bundle of the fiber. Endoneurium, connective tissue sheath around individual nerve fibers. This picture showing covering of connective tissue surrounding the nerve fiber. Endoneurium, connective tissue sheath around nerve fiber. Perineurium, connective tissue sheath surrounding the nerve bundle. Epineurium, the connective tissue sheath surrounding the whole nerve trunk. Many axons which form nerve fiber, many nerve fiber form a bundle of the nerve fiber, many bundle of the nerve fiber which form nerve trunk. Nerve fibers may be naked or ensheathed. Nerve fiber may be naked or encased by one or two sheaths. Each nerve fiber is formed of three structures: axis cylinder, axon, myelin sheath, neurolimmal sheath. Again, nerve fibers are naked or encased by one or two sheaths. Each nerve fiber is formed from three structures: axon, myelin sheath, neurolimmal sheath. Axon, it has cytoplasm called axoplasm surrounded by cell membrane is called axolemma. Again, the axon, it has cytoplasm called axoplasm surrounded by cell membrane axolemma. It is formed of three regions: axon, hillock, site of axon emergence from the cell body. It has no nasal substance. Two part or two region of axon is called initial segment. Initial segment naked, no myelin, no neurolimmal sheath. Three part or three region of axon is called axon proper. Again, the axon has cytoplasm called axoplasm surrounded by cell membrane called axolemma. The axon is formed of three regions: axon, hillock, initial segment, axon proper. Axon hillock, site of axon emergence from the cell body. It has no nasal substance. Initial segment naked, no myelin, no neurolimmal sheath. Three part is axon proper. This picture showing neuron, cell body, and axon and dendrites. This picture showing axon hillock. This picture showing axon and surrounding by myelin sheath and covered by Schwann cells. Neurolimmal sheath is a cellular tubular sheath around the axon, surrounding myelin F%. Schwann cell with its basal lamina, small flattened cells with few organelles, one cell corresponding to one internodal segment. What is the function of neurolimmal sheath? 1. Insulation. 2. Formation of myelin sheath of one internodal segment. 3. Nerve regeneration in BNS. Again, neurolimmal sheath is a cellular tubular sheath. A cellular tubular sheath around the axon, surrounding myelin sheath F%. Schwann cells with its basal lamina, small. What is the Schwann cells? Schwann cells is a small flattened cells with few organelles, one cells corresponding to one internodal segment. What is the function of neurolimmal sheath? Urolimal sheath has insulation and the formation of myelin sheath of one internodal segment. Nerve regeneration in peripheral nervous system. Myelin sheath. Myelin sheath is fatty tube that covers the axon proper. Myelin sheath is interrupted sheath at node of Renify. It is formed of several spiral layers of cell membrane, jelly rolling theory of Schwann cells and processes of oligodendrocyte cells. What is the function of myelin sheath? Myelin sheath is protective and nutrition. 2. Acceleration of the nerve impulse. 3. Insulation. Again, myelin sheath is fatty tube that cover the axon proper. Myelin sheath is interrupted sheath at node of radii. Is formed of several spiral layers of cell membrane, jelly roll theory of Schwann cells and processes of oligodendrocyte cells. What is the function? Functional: 1. Protective and nutrition. 2. Acceleration of nerve impulse. 3. Insulation. This picture showing myelination of a peripheral axon, Schwann cells and jelly rolling theory. Axon, Schwann cell body. This picture showing jelly rolling theory. Myelin sheath with light microscope, H&E not seen. Myelin sheath with H&E not seen because myelin sheath is fatty with dissolve during staining. Another with osmic acid. Myelin sheath appear with black circle. Again, myelin sheath with hematoxylin and leucine not seen due to dissolve of myelin fatty layer, appear vacuolated. Another with osmic acid stain, myelin sheath appear as black circle. This picture showing nerve fiber with electron microscope, myelin sheath, major dense line and intra-period lines. Major dense line, fusion of inner layer of cell membrane of Schwann cells and oligodendrocyte oligodendrocyte cells. Intra-period lines. Close contact of outer layer of cell membrane of Schwann cells and the oligodendrocyte cells. Again, with EM electron microscope, myelin sheaths appear major dense line or intra-period line. Major dense line, fusion of inner layer of cell membrane of Schwann cells and oligodendrocyte cells. Intra-period line, close contact of outer layer of cell membrane of Schwann cells and oligodendrocyte cells. Medical application: multiple sclerosis, MS. The myelin sheaths surrounding the axon are degenerated by autoimmune mechanism. In multiple sclerosis, the destructive action exceeds the capacity of the oligodendrocytes to produce and repair myelin. Again, multiple sclerosis is defect in synthesis and repair of myelin by autoimmune mechanism. Nerve fiber. According to myelin sheath, nerve fiber are classified to myelinated with neurolemma in peripheral nerve, non-myelinated with neurolemma in post-ganglionic sympathetic fiber, myelinated without neurolemma in nerve fiber of white matter of CNS, non-myelinated without neurolemma nerve fiber of gray matter of CNS. Again, according to myelin sheath, nerve fiber are myelinated with neurolemma in peripheral nerve, non-myelinated with neurolemma in post-ganglionic sympathetic fiber, myelinated without neurolemma nerve fiber of white matter of CNS, non-myelinated without neurolemma nerve fiber of gray matter of CNS. This picture showing nerve fiber classification according to myelin fiber surrounding: myelinated nerve fiber, unmyelinated nerve fiber, myelinated without neurolemma, non-myelinated without neurolemma. Nerve fiber connective tissue covering of the nerve trunk are endoneurium, perineurium, and epineurium. Endoneurium, connective tissue sheath around individual nerve fiber. Perineurium, connective tissue sheath around each bundle of the fiber. Epineurium, connective tissue sheath surrounding the whole nerve trunk. Again, connective tissue covering of a nerve trunk are endoneurium, which is surrounding the nerve fiber, individual nerve fiber. Perineurium, connective tissue sheath around each bundle of the fiber. Epineurium, connective tissue sheath surrounding the whole nerve trunk. Thank you.